Again, thank you everyone for joining. I will just, in my part of the presentation, go over a little bit of what you will learn in this particular program, Introduction to Bioinformatics, discuss some of the technical steps that you will be able to complete after you learn about the different types of omics data, and show you a little bit of the hands-on how you will use the learning to actually start doing bioinformatics as you prepare, hopefully, for more in-depth learning that will be covered in tomorrow's webinar on data science, and the webinar for orientation to the Transcriptomics program, which will happen on Friday. Let me go ahead and start sharing my screen. So Introduction to Bioinformatics is really a high-level program that is designed to introduce you to the key elements of computational or information technologies and biology, or the study of living organisms. And one of the kind of interesting things I think about bioinformatics is that it really provides an intersection of informatics and how to manage, analyze, process data, statistical analysis, including finding patterns in noisy data, and then biological interpretation. And so all of these elements are going to be linked together in a series of these asynchronous modules that you will be able to complete over this semester. Now, the applications of bioinformatics have really tremendously impacted different aspects of research, biotechnology, and translational biomedical research. And so what you will learn in this program will provide you with some insight into how that has been changing over the past maybe decade or two, as the complexity of data has really transformed the need for higher and higher computational complexity and analytical complexity to extract meaningful patterns from big data. In a lot of cases, this has led to many approaches to different types of treatments and diagnostic assays, as well as design strategies for experimentation. So what we will try to do in this particular program is kind of talk a little bit about how a biologist or somebody with more of a life science background can really start looking at data and learn how to process, analyze, and interpret some of the patterns that are found in this kind of data. Now, what kind of data are we going to be talking about? Primarily, excuse me, primarily we're going to be talking about next generation sequencing data. Now, next generation sequencing data could be used to generate many different types of omics data. This can include phenomics. Next generation sequencing is typically associated with a phenotype, and that phenotype could be described by how cells, tissues, and organisms function or not function properly in the case of disease. It will also talk about some of the elements like genomics, epigenomics, transcriptomics, and metagenomics. And so a lot of these are data sets that are publicly available in various repositories. For example, you can find large quantities of research projects, high quality data available through the National Center of Biotechnology, or NCBI, including repositories like GEO, Gene Expression Omnibus, SRA, Sequence Read Archive, and others, where you will have a design of the experiment or how the data was collected, together with access to publicly available data sets. Now, there are over 80,000 publicly available data sets just on GEO, which is processed transcriptomic data. And so to prepare the students and the faculty to make the most out of these publicly available resources and data sets, this program will provide you a good overview of the different options that are out there, different data sets that you can get access to, and tools that you can use to process and prepare this data for analysis or use in your own independent research project. So the Introduction to Bioinformatics program will cover the following topics. We will talk a little bit about bioinformatics and big data, or how these technologies have transformed the use of data analysis to understand biological phenomena. We will talk about genomics, or the study of DNA variation. We will talk about transcriptomics, or how this next generation sequencing technology could be used to analyze mRNA abundance and as a proxy to protein that is being translated from DNA. We will also talk about some of the statistical analysis and machine learning. So you will get a sense of how the noise in the data, the signal in the data, the large quantity and variability of data are all leading the way into more and more complex and sophisticated analytical methods. Now, don't be intimidated by this. This will typically be reduced to, what anyone can try and use without any in-depth understanding of a lot of these methods, just as a form of introduction. We will talk a little bit about scripting. So we want to provide you with some sense of what goes into a typical bioinformatics project, and that might include using user-friendly tools, which I will demonstrate today in just a few moments. It will also introduce you to some of the commonly used packages, so you understand not just the black box of a button that you press and get an output, but you really understand what goes on behind the scenes and how this type of process is conducted behind the scenes. And then finally, you will be introduced to some of the example projects. And so the example projects are going to provide you with a variety of different applications. You do not have to complete any one of those projects. This is just to provide you with a sense of what kinds of projects could be done and how do experts that publish using bioinformatics utilize different bioinformatics tools to extract meaningful insights. So as already mentioned by Lane, you will start this journey on this portal, learnomicslogic.com, which seeks to connect all the different elements of what I just described. So it will provide you with the coursework, the assignments, the evaluations that we will use in this program, as well as access to the different types of data sets and practical tools that we will utilize. Another simple way to find the right resource on this page is to go and take a look at some of the offered coursework. Now, anyone that registered for this program has access to a variety of different types of resources that I want to show you. So this is where you probably registered for the program. And I just want to show briefly that you can find on this page everything that we are discussing today in today's webinar. So you will find here the schedule that already was covered by Lane. You will also see here the links to registration. If you are confused with this iLab registration or you're not from LSU or LBRN, it's sufficient to just register through this form and we will actually reach out to you and confirm what exactly you're interested in. But you will also find here some of the modules that are included. So what I want to do is I want to just show you a little bit how to find these modules once you have created your account. So before I go on, maybe you could let me know in the chat box if you have already completed your registration on the Learn portal. So if you have, and I will just pause here for just a couple of minutes. If you already have an account, please put number 1 in the chat box so that I know that you have completed your registration. Excellent. Now, if you have already completed your registration, you have an account, I will ask Sonalika to now launch a poll just so that we have a sense of who's in this webinar today and where you're coming from. So Sonalika, could you please launch the poll? Okay, so it looks like the poll is not available. Let me actually change something real quick. Okay, can you launch it now, Sonalika?
Yeah, I think you can go ahead. I can't see the poll option. Maybe I need to re-log in with the account and then I'll just launch. Thank you.
Okay, so we'll continue and then towards the end. So if all of you, if all of you have already registered and you go to this courses area, you will find here several different things that you can do. So first of all, If you just joined us, I see a question there in the chat. You just simply go to this portal and you register for free. First of all, you create an account. And once you have an account, we started going through some of the specific courses that are included in this Introduction to Bioinformatics course and how to find them. So you can find them in multiple different places. And we have heard a lot of questions about where exactly to find all of the curriculum, what goes first, what goes next. The first area that I explained just briefly is that you have the central registration page. On this registration page, it is explained what the different options are, all of the different released videos that you will be able to watch asynchronously. And then down at the bottom, you will see all the different modules that are included, including here, you will see some of the people that have completed some of these and what they have gotten out of these modules down at the bottom. Now, you also have your page for the program where you can find all of the sessions that you can complete. That will be available to you only after you have registered for the program. But as you complete these, and maybe you want to go deeper, you will find here different areas that you can search. For example, if you're looking for more in-depth view of, let's say, transcriptomics, you can find the course right here. If you're just getting started and want to understand what is going on in bioinformatics in general, maybe search for Introduction to Bioinformatics. There are also different collections of courses right here. So we are with the Getting Started collection, and under the Getting Started collection, you will get all of the beginner level courses that include Introduction to Bioinformatics, a course in Python and R, just to kind of get you started with coding. You will also see a course called Bytes and Molecules, where you will see a lot of the information that we will cover in this program, including the terminology about different types of omics data and the different types of biological experiments that could be conducted and data could be generated to study different phenomena. You will also have some introductory courses specifically to genomics, transcriptomics, and we're adding more and more resources into this course to make sure that anyone who's getting started has the terminology and kind of a high level overview of what all of these different elements are, what do they mean and how are they used. So you will also find that under this specific Getting Started link, and I will share the link right here too, there's a course called Designing a Bioinformatics Research Project. So Lane already mentioned this, but you can essentially learn about these data sets and about the different use cases. But we hope that you take this further and you're able to apply your own research interest to the areas that are covered by bioinformatics. So you can identify a project, a data set, and the research question, and essentially, if you are interested in that kind of a component, especially if you're a faculty or a graduate student, then we welcome you to reach out to us additionally so that we can explain how to best leverage that in the context of this introductory program. So now that all of you are registered, I see a lot of people have put number 1. So I asked previously that if you are registering, put number 1 in the chat if you have completed your profile. Again, once you're there, you can click right here on your profile. And like Lane already showed, you will see your profile with all of the information that you need about the different programs that you're a part of, etc. All right, excellent. So let's go to this Introduction to Bioinformatics program. Okay, let me just go to programs. And I want to show you where you can have access to our mentors that will be available throughout the program for any questions that you have, right? So an important part of this is that you're not on your own. You're not just doing this completely asynchronously and have no one to talk to. Once you're a part of this program, you will be able to scroll down and you will see that there's an option to ask a question. So to ask a question, you will be redirected to the Slack channel. And in the Slack channel, you can communicate with other participants of the program or with our team. And so our team is going to be monitoring this. We're not going to, promise you that we will answer all the questions within a certain time frame, but we will be monitoring this introduction to bioinformatics program. You can see that some of you have already been added. If you have not been added to this program, please go ahead and click on that link on that page. And you can just say hello, and that way we can start getting connected and kind of getting to know you through Slack. Now, you also have on this page, if you have more in-depth questions, so especially if you're confused, maybe you feel frustrated with something's not working as you're expected, you also have here an option to book a meeting. And so if you want to have a meeting with someone from our team, you will see here a link to schedule a meeting with Sonalika, who's also on this call, so that you can ask specific questions. Maybe technically you don't have access to some part of your account. Maybe you're stuck, you don't know how to move forward. Maybe something is not working as expected, right? So first you can talk to Sonalika. She will try to help you as much as she can. If she can't, she will also connect you to one of us, Dr. Harpreet Kaur, who has a PhD in bioinformatics. If you are doing this online, not from LBRN or LSU. If you are from LBRN or LSU, especially if you're a graduate student or faculty, we're also connected with the faculty from LSU and from LBRN, and we would be able to connect you to the right person to answer your question. This program is a part of a whole network, and so you will see that there are a number of different resources, as already mentioned by Lane, that you have access to, including labs, support, technical support, and advice from experts as well. So please make sure that you utilize these options as well if you are joining this program. Now, another part that I wanted to share today is that together with your account, you get an access to the T-Bioinfo platform. And so I want to spend the next couple of minutes just briefly explaining what this platform is and how it's going to be utilized by those of you that might not have any bioinformatics background at all. Once you have registered, so again, this is something that might take a little bit of time because maybe you've registered on iLab, maybe you directly registered through the portal. However you register for this program, this is going to be a part of the subscription. So you will have some level of access to this platform. And what I will show you today is just an example of a high-level overview of what is possible in this specific program. So as I mentioned, there will be an overview of different types of omics data, including genomic data, transcriptomic data, metagenomic data, so that you have a sense of what next generation sequencing data looks like and what to do with this kind of data. So first of all, in this areas of analysis section, you can hover over each different section and learn about different types of methods to produce this kind of data. As you go inside each section, for example, we're going to be focused on next generation sequencing data, you will see that right next to each section, there's also this eye. If you hover over, it will tell you a little bit more about what this section does. Now, for those of you that are just registering, you have a demo account. The demo account is designed to show you very quickly how the data was processed and what is the typical output. So to explain, I will show you a couple of sections today so that you have a good sense of what we're going to be doing. If you scroll down to RNA-seq chip and you open this drop-down button, you will see that several exemplar projects are going to open up. And the first one is going to be this RNA-seq cell line project. So we can go ahead and click there. Once you click here, you will see an interface that essentially represents different types of methods with a visual button. And the ones that you can press are highlighted with this yellow line. So for example, start is highlighted. So in the context of RNA-seq, essentially these pop-ups will guide you to explain what each button does. And when you go through those sessions that are linked, for example, to transcriptomic data, we will go in depth into an overview of what each one of these buttons does and how to use them including how to build different types of pipelines for different objectives or different research questions that you might have in mind. Here you will see the typical steps for an RNA-seq pipeline, which includes pre-processing, mapping, and quantification. And if you scroll down, it will also show you how to build such a pipeline. So let's go ahead and build it. Once I click on OK, you will see that other buttons are now highlighted, and I can select from a number of different paths to build my pipeline. Each path will give me a different types of an output. For example, I can start by pre-processing the data with Trimomatic, PCR clean. Again, once we get into this module, you will understand a lot more of what each step does. And after that, you can see that I can click on Bowtie2T, which is a method of mapping the individual reads to the reference genome. And then I can quantify the level of expression by using RSEM, or RNA-seq, using expectation maximization. The objective of this step is to quantify, to give me a number for each gene that I have in my table in the annotation, and each sample, which is where my data came from biological samples. If I click on OK, I will be able to click on End. There's more information, of course, here that you can learn about, but in the output files, you will see a typical output of this type of a pipeline. So the objective will be to really understand how a pipeline or a combination of multiple scripts and algorithms can take raw data that we put in at the beginning and produce some outputs that we can explore right here. Now, after you've understood this basic concept with something like RNA-seq, you can now go back to areas of analysis and see similar examples for other types of data. And so another type of data that I want to briefly show you today is this processing of metagenomic amplicon sequencing, which allows us to understand diversity of ecological microbiological samples directly without culturing the specific bacteria that we would like to study. So again, you will have an introduction to metagenomics in this introduction to bioinformatics course. You will see a little bit about how this data is prepared. You will also learn about specific methods. For example, the data to pipeline includes several different steps that you will need to understand as you try to analyze your data, metagenomic sequencing data, and then how to interpret this data as well. So what is the significance of different steps? What do these steps actually do? And how do you combine these steps into a workflow to ultimately get a result that you can critically evaluate to understand what it means from a biological perspective? Now, sometimes you will have tables, sometimes you will have visual outputs. For example, here we can download this PDF file and see the output from this pipeline provides us with a sense of diversity and different types of analyses that we can ultimately perform to kind of understand the differences between different types of samples that were included in this particular project. So again, each pipeline represents a project, a specific data set, right? So each one of these demo pipelines represents a different project example and shows you how you can get from raw data to processed data that you can interpret or perform downstream analysis. Now, as already mentioned, we will cover a little bit of everything. So if you go back to your program page, you will see here the schedule will give you a sense of different methods, different types of omics data, statistical analysis of omics data, including the introduction to biomedical data science. And so here with Introduction to Biomedical Data Science, you will have access to this course. The course covers many different steps for exploratory data analysis, statistical analysis of data, including machine learning that you will be able to practice on the T-Bioinfo platform in the context of this particular program. Okay, let me just stop sharing real quick so that this loads. So in the context of this particular program, you will find this course as a user-friendly, again, series of pipelines, including some example data sets. And then if you're interested to learn more, we have 2 other programs that are going to be introduced tomorrow for data science, which is going to be an in-depth view of coding, statistical methods, comparison and benchmarking of different types of machine learning methods, including deep learning, and the application of these kinds of methods to different types of large data sets. And on Friday, another orientation session that will be dedicated to in-depth transcriptomic data analysis. So briefly, I want to show you those courses as well. Before I do, are there any questions about what we covered today? Okay, so if there are no questions, let me show you a little bit about these upcoming courses. The one that we will do tomorrow, which is called Data Science for Biomedical Data. Again, this is sponsored by LBRN and Biomed. You can see right here, the orientation session is tomorrow, October 7th. And you will see a little bit more about the different topics that will be covered. So we'll talk a lot more in depth about the nature of these kinds of data sets and what to do with them, starting from visualization to exploratory analysis, to statistical analysis for hypothesis testing, to eventually machine learning and deep learning as well. One of the differences between this introductory program and the in-depth programs is that the level of complexity will increase. The number of hours that you're expected to spend working on this program, the different assignments that you will get, will also increase. And so we have an equivalent to kind of the academic standard. The introduction to bioinformatics is about 1 credit course training module. The data science is about a 2 credit, an equivalent of a 2 credit course training module. And then the last one, which is going to be the transcriptomics one, is an equivalent of a 3 credit course module. So here you will find also the link to the transcriptomics module. And I will just paste these links here as well. So this is going to be an orientation session starting on Friday. The previous course for data science is going to be tomorrow. So make sure that you join if that is interesting to you. And all of these are asynchronous. So essentially you can start learning already. After each session, do not forget to click on complete the course. So here, if we go back to our programs, you will find that under each session, you can complete that session, and that, number 1, gives you points. Number 2, ultimately, after you complete all of these sessions, if you scroll down, you will see that your name and the complete certificate will be posted once you have completed all of these sessions. And they're structured in such a way that you should have plenty of time to go through all of these materials.